Good morning, everyone. My name is Anshita Raghuvanshi, Senior Assistant Professor, School of Pharmacy, BBTU, Lucknow. Today, here I'll discuss about the topic granularity of TT process, that is technology transfer process, and some parts which I'll discuss here is active pharmaceutical ingredients and excipients. Starting material. The specification of starting material, that is APIs and excipient, active pharmaceutical ingredients and excipients to be used at receiving unit should be consistent with reference batches. That are reference batches are our development batches which were developed at sending unit or from where the technology is being transferred, bio batches or batches manufactured at sending unit. Like all the data should be similar. The quantity which were used there, the in sending unit and the quantity which are being used here where we are implementing our technology in receiving unit, the batches, data, amount, quantity, everything should be consistent or should be matching according to the data in sending unit. Any property which are likely to influence the process or product should be identified and characterized. If there are some changes or if any property which can influence our process or production should be first identified and characterized so that we can resolve it and continue our smooth process. The material is active pharmaceutical ingredient. The sending unit should provide the drug master file, that is DMF, and any relevant additional information on the API to receiving unit to be checked against the specification of API. When we transfer our technology from sending unit to receiving unit, the receiving unit should provide every important data, every important specification of the drug or uh, material in a form of file which is known as drug master file, in which everything will be specified. Any information, any minute information, major information which is to be checked at every different points, everything is mentioned in drug master file. All the specification of API is mentioned there. So the basic information which our drug master file contains are: number one, the information of the manufacturer, from where the active pharmaceutical ingredients is being manufactured, who is the manufacturer of API. Next, flow chart of synthetic pathway outlining the process, including entry points of raw material, critical step, process control, and intermediates. Here, like how our it should contain a flow chart which will denote that how what is the synthetic pathway, how this API is being manufactured, what is the process of manufacturing the API, outlining the process, how the entry points of raw material, like from which gate, which type, which method the raw materials will enter, what are the critical steps which we will monitor. So all these things is being denoted in API. It should be similar according to the sending unit, and same process should be followed in receiving unit. Definite form, like if our API contains any photomicrographs and other relevant data, any polymorphic and solvent form, like if API is convertible or it will by how we will protect our API because there are some changes because of it, API will changes properties. So all these points should be covered there. Solubility profile, in which type of it is solvent the our drug or main active pharmaceutical ingredient is soluble. What is the partition coefficient and how we will determine the partition coefficient, method for the determination of partition coefficient. Every basic thing is included in drug master file. Next is intrinsic dissolution rate. Like what are the methods, how we will determine the dissolution of the drug, particle size and distribution, bulk physical properties, including data on bulk and tap density, all the things which we perform in lab, basic data, basic initial testing data, or everything is mentioned in drug master file. Water content, hygroscopicity, how we'll determine the water content, in microbiological consideration like sterility, bacterial endotoxin test, bioburden levels, where the API supports microbiological growth in accordance with regional pharmacopoeia requirement. Specification and justification for release and end of life limits, summary of stability studies conducted in conformity with current guidelines, including conclusion and recommendation on retest date. The method how which we have conducted the stability studies and what are the results of stability study. We the sending unit gives the retest date. Retest date should be mentioned there so that we can perform retest that is our drug still stable or when it will get expire. So all these things have is to be done in receiving unit, and all this data is contained in drug master file. Listing of potential and observed synthetic impurities with data to support proposed specification and typically observed levels. Next is information on degradants with a listing of potential and observed degradation products and data to support proposed specification and typically observed levels. Like if our API has some degradation, how it get degraded, small or it is a very important information which should be provided in drug master file. Like if I am observing this type of degradation, then it should be mentioned whether it is observed there also or not. Okay. So these all the information regarding active pharmaceutical ingredients should be given in drug master file. Next, we will start with excipients. Second and most important, excipients. Like information of API is very important, same the information regarding excipients is also very important because this is also a very, very important part of any manufacturing process. The excipient to be used have a potential impact on final product. Their specification as well as DMF should therefore be made available by the sending unit for transfer to the receiving unit or site. The following information should be provided for all types of excipients. Number one is description of functionality with justification for inclusion of any antioxidant, preservative, or any excipients above recommended guidelines. Like if we want to include any excipients more than the given limit, then we have to give the justification why this is required and what is the use of using extra in amount. Okay, next is the manufacturer where the excipients is manufactured. Next, all the specification like monograph, additional information that may affect product processing quality for compendia excipients, or a complete listing of specification including analytical methods and justification for release limit for non-compendial excipients, for excipients used for the first time in human drug product or by a new route of administration. The same level of details as for drug substance should be provided. Special consideration with implications for storage and or handling, including but not limited to safety and environmental factors. Example, as specified in material safety data sheet, and sensitivity to heat, light, or moisture solubility. Like if our ingredient is light sensitive or it is a hygroscopic, moisture containing, sensitive to moisture, soluble, easily soluble, sensitive to light or heat, then all these types of information, safety and protection regarding information should be given in DMF so that receiving unit should consider it and do as appropriate. Regularity consideration, that is compendial status and appropriate regularity information for non-compendial excipients, information on residual solvents or organic volatile impurities, and documentation to support compliance with transmissible animal spongiform encephalopathy certification requirement wherever it is applicable. Thank you.